Thank you very much for inviting me here to talk about this work. And thanks for you, most of all, to stick around for a talk that is clearly not entirely about diabetes. So most of what I'm actually going to talk about has to do with Neanderthals. And so what I would like to do at the very start of the talk is to introduce you to Neanderthals, what they are, how they were discovered, and also tell you a little bit about the genetics of Neanderthals. And so to start really from scratch, so Neanderthals were first discovered and recognized as something different from us in 1856, when they were discovered in the Neander Valley in Germany, which is where they get their name from. And already in this kind of, shortly after these bones were actually discovered and this skull fragment was discovered, it was actually given to researchers to look at. And one of them, for the time, made almost immediately this conclusion that this particular bones belong to an individual that is from some very old group of humans. So in loosely translated from this document from 1858, he says that this is kind of one of the. No? Okay, too quiet. Okay, is that better? Good. Alrighty, or I just speak a little bit louder. I can do that too. Alrighty, so. So the description essentially says that this might actually be the oldest remnant of what we actually, one of the oldest remnants of some earlier inhabitants of Europe. And this conclusion he came to by actually looking at the bones in the skull, and he says that they actually contain certain features that make them look as if they came from a particular ancestral version of humans. And this is quite impressive, given that this is essentially before the origin of species from Darwin. So his interpretation didn't really catch on right away. It took several decades before people actually came around really understanding what he meant to say, and that this is really something special and something very old, and not actually just a normal other human being or a diseased human being or something like this. But also in the last 150 years, since this discovery, our understanding of how Neanderthals were and how they actually behaved changed quite a bit from anthropology and archaeological findings. And so what you see here is a reconstruction of a Neanderthal on the left-hand side and a modern human on the right-hand side. And these two, so our early ancestors and Neanderthals, were very similar in many respects. So one of them is that they lived in small groups, that they were hunter and gatherers, that they used stone tools at the time, and there's also evidence from fractures that they gotten that were healed, that they had some understanding of treating wounds, and that they actually cared for their sick. There are some evidence that there are burial rituals or burials actually of Neanderthals. And so in many respects, the things that you would actually think of when you think about our own ancestors, many of those also apply for Neanderthals. But despite all these similarities, there was also something that kind of made them remarkably different, and that can be seen, for instance, in the shape of the skull. So again, on the left-hand side, you see a skull of a Neanderthal. On the right-hand side, you see a skull of a modern human. And despite the fact that this picture is a little bit stretched, one of the things that one almost immediately notices is that the Neanderthal skull looks a little bit more elongated and a little bit flatter than the more round skull that you actually see on the right-hand side of a modern human, so our own ancestors. And this is one of the fairly interesting features. There are other features that one can actually find when one looks at the skeletal morphology of Neanderthals and modern humans. One thing that I know Ola is very interested in are the attachments of the muscles on the bones that gives some hint that Neanderthals were substantially more muscular than modern humans are on average. So the attachments are actually much thicker, indicating that there were more muscles. And so this might actually be a specific feature of Neanderthals that we do not share. So one of the things that we would like to do is to understand where these differences come from genetically. And so how we can actually go after this is by studying the DNA of Neanderthals. And very briefly, so how this is done in Leipzig in the lab of Svante Pääbo is by taking bones that researchers give to us, so after an excavation or people that actually curate collections of bones, then a small hole is drilled in these bones to actually collect bone powder, and this bone powder is then used to extract DNA. All of this we try to do in as clean of conditions as possible for the reason that it is very important to avoid contamination from modern humans, either through labware or through the handling of the bones. And so all of this is done in clean room conditions. On the top right-hand side, you see kind of one of the facilities. What doesn't help against contamination is to actually turn the light off in the lab. This is really just for the camera so that it looks good. So despite all this effort to excavate, to get to this DNA, it quite often doesn't happen. And even if you actually get DNA out of a Neanderthal bone, it has specific features. Actually, all old DNA has specific features. And so one of the features that you have to deal with is that over the time, DNA breaks down into short pieces. And what we often see is a length of 50 to 70 base pairs on average for the fragments that we extract. They can also be much shorter if the sample is older or more degraded. This is here just an example of a distribution, so of course not all fragments are the same size. It's a distribution. Another issue we have to deal with when we work with ancient DNA is ancient DNA damage. And so the damage that is most prevalent is cytosine deamination, meaning that you form a uracil from cytosine, and this will then be misread by polymerases as thymines, so you get C to T exchanges. Those will actually accumulate preferentially at the ends of molecules because this process of cytosine deamination progresses faster if you're in a single-stranded bit, which quite often happens at the ends of the fragments because they don't break off completely flush, but there's a little bit of an overhang. And so the biggest issue that we are actually facing when we are looking for Neanderthal DNA is that not all the DNA that we can actually get out of a bone really comes from a Neanderthal. Most of that will actually be from different bacteria or other environmental sequences where this bone was actually lying. And so quite often what we see is that we have just 1% or a couple of percent of the fragments is really coming from the individual that this bone belonged to, and the overwhelming majority of the sequences we get out are some other contaminants. As with all of these general rules, there are exceptions, and one of these exceptions actually comes in the form of a cave in Russia called the Denisova Cave in the Altai Mountains. So this cave has year-round fairly cold temperature, and this and other features of the cave seem to actually help so that some of the material that is found there is actually extraordinarily well preserved. And so two bones have actually been excavated there that contain a lot of DNA that can actually be used then with the help of new laboratory methods to generate entire genomes from and fairly complete genomes from archaic humans. So one is a small finger bone and the other one is a toe bone. And due to this extraordinary preservation that actually so about 70% of the fragments that were actually from these bones really were from these archaic individuals. We were then able to sequence them to 30 and 50-fold coverage respectively. A little while later, we then also added to this another genome from the Vindia cave, which is in Croatia, another 30-fold coverage, and that was from a bone that had at least 10% of the fragments coming from a Neanderthal. So this was allowed also by the progression of the sequencing technology, which gets, of course, cheaper all the time, so that we can actually afford to sequence this deep enough to generate an entire genome from it. So these are the archaic genomes that are currently publicly available. And when you look at the relationship of these different genomes, one thing that you actually see is that the bone from the Altai Mountains, so the bone from the Denisova cave, actually matches most closely with the Windia, with other bones from Windia. And the finger bone, the small finger bone, is actually from a group that is more distantly related from other Neanderthals, but still is a sister group compared to us modern humans. So with these sequences, one of the things that one can actually go after is to try to understand the genetics of uniquely modern human traits. And this really goes back to the title of this talk. So what I'm trying to go after here is really these ancient phenotypes that developed sometime in the past and then set us apart from Neanderthals. And so one case in point is this slide that I want to return to very briefly, where we see this skull morphology as a difference between Neanderthals and modern humans. And the skull that you see on the left-hand side, this different shape, is actually something that resembles more closely the skull morphology that one sees, for instance, in chimpanzees. So this might actually be something that specifically evolved in our ancestors, how this skull actually looks like. And so if you are asking for specific changes in this time frame, so something that is common to all of us but different from Neanderthals, then from our estimates we can say that this would be substitutions that happen sometime around half a million years ago up to 100,000 years ago, when different modern populations actually separated. So somewhere in this schematic, somewhere on this red lineage. And so one way to go after this is then to just go and ask for any kind of change that you can see in modern humans that are different from those in Neanderthal and chimpanzee and many other groups that one can use as an outcome. And so when you do this type of analysis, what you see is that there are a large number, several tens of thousands of nucleotide changes that actually correspond to this description. So they seem to be specific for modern humans, and we all have those compared to Neanderthals and others. A smaller number are insertion deletions. There are some that actually change the protein. And so since these are particularly interesting, one of the things that is going on in Leipzig is to study this set of just 96 amino acid changes specifically. But there are also synonymous changes in proteins and so on. And so 30,000 or 35,000 changes is quite a lot of changes to study immediately and directly with any kind of laboratory technique. So this is a little bit too much. So one of the things that one can do is either restrict yourself to amino acid changes or some changes that you think might have a functional consequence. And the other way to approach this would be to actually look for signals that tell you that these changes might have been beneficial for our own ancestors in the past. And so I will talk a little bit about the second part. So there are a number of different tests that one can actually apply to data if one has different genomes from modern humans by comparing them to look for specific signals that tell you that selection actually acted on a specific variant or a certain region that contains variants. And so in this slide here is from a review that is already a couple of years old. There's a nice summary of the different tests that one can actually apply. Some of them actually work better in recent times and some actually work better in more distant times. So unfortunately, the specific time frame for Neanderthal-specific changes falls in an area where neither the long-term tests already have enough power to actually detect selection, nor the short-term tests are actually really geared towards finding out whether selection happened. So we had to think about how we can actually find evidence for selection. And one of the things that one can think about is then what type of signal do we expect? And so often these tests for selection, they start with a model, and so the predominant model is that of a classic selective sweep. And what that says is that at the start of selection, you have a mutation that occurs. This mutation is immediately advantageous for the individuals that carry this mutation. So in evolutionary terms, this means they have more offspring or they have a higher chance of offspring. And so what this leads to after a certain time frame is that more and more individuals will carry this particular variant. And since it is an advantage to carry this mutation, you will drag along a certain piece of sequence around it that will not be broken down by recombination as fast as neutral variants are. So you will kind of form an area around this mutation where the sequences look very much identical or very closely related at least. And the archaic human in this model would not be affected by this. We are asking for mutations that are modern human specific. And so if you think about this in terms of how a tree would look like, one of the things that you would see is that for this particular region where you actually had positive selection acting on modern humans, you would find a phylogenetic arrangement where the archaic human, be it Neanderthal or be it a Denisovan, would fall outside of the variation of modern humans. And that is really just because all the modern humans would actually look identical at the particular position where selection happened and also surrounding it. And now this will only help for the construction of a test if it would actually be different from what you would normally expect to see over the genome. And so one thing that one can do is actually just simulate the genome with the parameters that we know from real data and just see how often would we actually expect these regions to occur. And the good news is we would actually expect in most cases, if we simulate a large number of modern humans, we would expect that the archaic human, so Neanderthal or Denisova, actually falls within the diversity. And it's really just because we look at many, many humans and there's always a chance to find one that is actually more ancestral. And if you actually find external regions, so where the archaic actually falls external, they usually tend to be quite short. And so this was then our starting point for implementing a method to find these old sweeps, old selective sweeps. And this is done by inferring local trees. So essentially how this works is we take the data from the 1,000 genomes, we go over it, either look at polymorphic sites or sites where all modern humans look the same, and then ask whether one particular lineage, let's say a Neanderthal, carries this new change or not. And this is our only information that we have to kind of infer this phylogenetic relationship at this particular location, so this local gene tree. And so a little bit more technical, how we implement this is as a Markov model with 3 different states. One for these internal regions where the lineage of the archaic falls inside the variation, and then 2 different ones, one for just normal external regions that just look as if they would be random, and then for the selective state shown here as S for those regions that are particularly long and that might actually come from selection. So we applied this then using the archaic human genomes to our data to actually find selection. And one of the first things that we wanted to find out is whether our test actually does better than other tests that have been invented before. And so these different tests that exist, they all have different powers for more recent time. So we would expect that if we take an estimate of the age of the region or the variants that we find in the region, we actually expect that our test finds older region. And so adding to this violin plot that just shows the distribution of the ages, there's a minimum age that is actually estimated here from variable sites in modern humans. We see that our test indeed finds older variants. So we can then apply this to all the different chromosomes to predict the regions and just look at how often do we see selection on which chromosome. And so when you make this comparison, one of the things that actually sticks out is chromosome X. So chromosome X has much more evidence of selection acting on it than you would expect just from the fact that it has a slightly lower effective population size due to the fact that it's just 3 chromosomes per generation that are segregating. And this is actually quite interesting because it matches a recent publication that found similar signals of selection that are quite common on chromosome X based on a completely different measure between modern humans and apes. and finds that selection seemed to have actually acted quite a lot, and the authors speculate, and by now have actually found more evidence for this hypothesis, that this is driven by ampliconic regions that actually influence whether the X or the Y chromosome is actually segregating in the germline. So you would actually veer off the ratio between the two different sexes in the population if you would actually have a specific variant in these regions, and that would be compensated in the long run. And interestingly, when we look at our sweeps that we find, we actually see that they are significantly overlapping with these particular regions. So it might be that not just in the ancestor with the apes you had evidence for selection for this, but you also find this since the time we separated from Neanderthals. Another global signal that one can look for is to see whether there is any specific overlap with functional regions. And so it would be, of course, very nice if you would find that genes or coding regions are particularly often overlapping. That would be an indication. We did not find any significant signal for that. But what we do actually find is that the regions that seem to have been under selection are more often overlapping enhancers and promoters. And that would indicate then that at least some of the phenotypes that were under selection might have been actually might have come from some regulatory changes. Just one candidate I wanted to mention is actually a pair of two different genes that reside on two different chromosomes, and that we found both of them. We found selective sweeps that are either overlapping them or are at least very close to them. And these two genes are actually interaction partners, and one of the genes plays a role in guiding exons in the nervous system. So there might actually be some role for the nervous system in those. I can only speculate what this essentially means for the differences between Neanderthals and modern humans, but it's quite interesting. So, so far I was just talking about how we can actually look at our own history and the particular changes that we experience, and whether they are positive or negative, whether they were positively selected. So how we can go about understanding our own phenotype that developed since we separated from Neanderthals. But there's also quite a bit that one can actually learn about Neanderthals themselves. And so one reason to do so is for the reason that for several hundreds of thousands of years they actually lived in different areas at different times in roughly this area depicted here in this map, so somewhere in Europe to Western Asia. And so they survive different climatic conditions, and it is quite interesting to ask how many Neanderthals were around and why did they actually disappear. And so for this first question, how many Neanderthals are around, one very basic measure that one can use is to actually study how the heterozygosity of an individual. So heterozygosity really just means that you take the two copies of the genome that an individual inherited from its mother and its father, and you just calculate the average number of differences. And so the logic is that if this mother and this father was randomly drawn from the population, then by just counting the differences, you get an idea of how different people were in this particular population. And so when you do that with a number of present-day Africans and present-day non-Africans, but also the archaics, what you see is that Africans have a difference of around 1 in 1,000, while non-Africans are somewhere in the range of 6 to 8 in 10,000, and archaics are much lower in the range of 2 in 10,000. So what is driving this lower heterozygosity in people outside of Africa is the out of Africa event, by which only a group of humans actually took out a small amount of the diversity, or a smaller amount of the diversity that existed in Africa as a whole. But it's very interesting to see that archaics are actually so low. And this is not just true for these two. There's also a third one that I mentioned earlier, the Vindhya genome, that is looking very similar. So one thing that one can do is actually study how this is actually distributed over the genome. And so one of the things that we noticed right away is that the Altai Neanderthal contains some very long stretches on its chromosomes on her chromosome that indicated something interesting is going on. And so these long stretches, they sum up to around 1/8 of the entire genome. And so from just family relationship, you can kind of put this into context by saying this would be roughly the similarity between you and a great-grandparent. And so similar to this idea, Flora Fay at the time in Berkeley actually used the data to make a comparison of different phylogenetic trees that could actually explain how these long stretches could actually appear. And you can pick now one of those scenarios that would explain this. One of the things that one could say is that they were related, the parents of this individual were related at the level of half siblings, for instance. And so by the same logic, so that you just take chunks and you look for chunks that have a certain size and a certain similarity, one can also make an estimate of population size over time. So instead of just looking for the last couple of generations, very long chunks, you can look for shorter chunks with a little bit more dissimilarity, and you just count how often those happened, and this will give you an indication of how many individuals were around at that time. And this is the logic behind a program called PSMC, or a method called PSMC. And what I show you here is just the estimate of the population size, or the effective population size of the Neanderthals and the Denisova compared to modern humans. And what you can see is that for the last 100,000 years, or even a bit longer than that, Neanderthals lived actually apparently in relatively small population sizes. So one thing that is of course very interesting for us, again, is to ask what did actually happen in terms of admixture with Neanderthals. And so this question is driven by the fact that modern humans, so our own ancestors, left Africa sometime around 100,000 years ago, and then went into Europe and Asia, maybe around 50 to 40,000 years ago, and settled in these areas. And so interestingly, if you take the last remains that you can find of Neanderthals in Europe, you actually find out that they essentially disappeared around 40,000 years ago. This is kind of interesting. And so one way to figure out whether some admixture happened or not is by doing an analysis that is called a side pattern analysis. Essentially how this works is you take a genome from a present-day non-African, genome of an African, a Neanderthal and an outgroup, and then instead of actually, so you line all these genomes up, and then you can take the basis, and you, instead of writing down the actual basis, you just write down what pattern you observe. You always label the last one, the outgroup, so chimpanzee, for instance, as an A, and any other base as a B, and then you just sum up how often you see which pattern. And this gives you then different counts, and two of those counts you can compare directly, and they are informative for whether or not admixture happened. And that's called a Patterson's D statistic, and these two patterns are BA-BA and AB-BA. And so BA-BA means that a derived mutation, a new mutation, is actually shared between a non-African and a Neanderthal, while AB-BA means that you actually have an African that is sharing a derived variant with a Neanderthal. And so if there is admixture between people that live outside of Africa and Neanderthals, you would expect that this count actually is higher than the other count. And this is exactly what we see, and one can actually normalize this to find, to get to an estimate of how much of the genome of people outside of Africa is actually derived from Neanderthals. And this percentage lies somewhere in the range of 2% to 2.5%. So for most people in the room, I guess, this would be essentially some Neanderthals in your ancestry. And so far this is just calculated based on just thinking of single sites. We don't really think about how these Neanderthal variants are actually distributed over the genome. And so to kind of understand that, one can actually make a little thought experiment and just go through it. So obviously you have, we are diploid organisms, so we have every chromosome twice. And one can actually label these by whether they come from the mother or the father. And then if you would actually label them by your grandparents, you would see that recombination happens in your parents that actually mixes up the chromosomes that you inherit from your grandparents. And of course, you can actually go on like this to the next generation, to the next generation, to the next generation. And what you see is that these chunks kind of go shorter and shorter and shorter. And so if you had a long time ago some Neanderthals among your ancestors, shown here in gray, what you would then expect if there was just a couple of ancestors from Neanderthals is that their ancestry actually pops up as small pieces among your chromosomes. So there will be short contiguous pieces that come from these Neanderthals. And so this logic here is that the longer ago that is, the shorter these pieces on your chromosomes from Neanderthals will be. But it also means in the other direction that if you actually look at a human that lived a long time ago, you would expect them to be larger. And so we were able to test this by looking at the 45,000-year-old modern human, so a human that lived in Western Siberia. And this individual was sequenced, and then we just labeled the individual variants that came from Neanderthals as either yellow or blue. And in this bottom plot, you actually see a number of different present-day humans: French, Sardinian, Han, Dai, Caritiana, and so on. And you see that there are these small yellow dashes that indicate that there are small pieces in this genome. But on the very top, you actually see this genome from this individual that is 45,000 years old, and you actually see fairly large stretches. And so based on these, that you actually see these larger stretches, you can definitely confirm that this admixture happened if there was any doubt. But what you can also do is you can actually date when this admixture happened, and that is 50 to 60,000 years before now. And so similar to this analysis here, just with a couple of individuals, you can also generate an entire map for the individuals in the 1,000 genomes, so the European and Asian populations in this particular plot, and then just look how often you have individuals Neanderthal ancestry over their genomes. And this was one of the analyses that Sriram Shankararaman did. And so we have now kind of this complete map of where people of the 1,000 genomes carry Neanderthal ancestry. So one of the things that you might ask yourself by now is what does that actually do? So it's interesting. So we had mixed with Neanderthals, we have a bit of Neanderthal, but what do I actually care about that? So to phrase this differently, what one would really like to know is if I have a certain piece of Neanderthal ancestry in me, is it actually good or is it bad or is it neutral? And so for the neutral part, what we can say already from the map that we produced is that we know that large fractions of the modern human genome or of our genomes are essentially oblivious to any changes. You can have any changes, it doesn't really matter. And you can actually reconstruct 40% or over 40% of a Neanderthal genome just by taking all the pieces of the different 1,000 genomes individuals. So it is reasonable to assume that there are large parts of Neanderthal ancestry that doesn't do anything, that is just present. But how about the negative side? Let's start with that one. So one of the things that one can do is to classify the genome into bins of how important these pieces are for the function of the organism, or in terms of selection, how much you try to eradicate kind of new mutations that might be deleterious. So this is captured in this B statistic, and you can classify this. And the lower this B statistic is, this bin that you see here, so 1 would actually be very, very close to genes or overlapping genes, while 5 would be very far away from genes, so very neutral. And so what you see is that generally the Neanderthal ancestry goes down the closer you get to genes and the closer you get to negatively selected regions. So this gives you some indication that some of the things that came from Neanderthals were kicked out, and they were especially kicked out if they overlapped something that might be functional. So another thing that is quite telling that there is something going on with Neanderthal ancestry in terms of negative selection is that you see large stretches in the genome where you have very little evidence to absolutely no evidence that anyone that in the 1,000 genomes actually carries Neanderthal ancestry. So it seems that there are these large chunks where whatever Neanderthal ancestry was there was kicked out. And so it's quite interesting how what mechanism could actually do something like this. And so as part of this paper of this map, of Neanderthal ancestry, we then looked at the expression of genes that fall within these regions, and we looked for an enrichment. And what we found is that the particular regions contain quite often genes that are highest expressed in testes. And so this indicates maybe that this has to do with fertility or some problem to have essentially kids with Neanderthals. And that would, of course, lead in the first couple of generations to these genes to be lost. And if you lose something in a couple of generations, it would affect a large region. People also have taken then these maps and tried to understand this entire process of kicking out particular variants a bit better. Since we know that Neanderthals lived in small populations over a long time, there's actually a chance that they accumulated some more negative alleles than our own ancestors did before this admixture. And so when they actually later on met, some of these alleles were actually kicked out. So there's some modeling going on where people try to understand this by building, by doing simulations and other processes to understand this. So this is all the negative part, but was some of that actually good for us? And so there's one really fantastic paper by Rasmus Nielsen and his group on one particular allele that has been found to be in frequency very different between Tibetans and other populations in Asia. And this particular allele is known to actually help with altitude adaptation. And when the group then later on analyzed this by comparing it against the archaic genomes, what they found is that this particular region that contains this variant matches very, very well to the Denisovanes. So it seems to actually come from Denisovanes. So what the model here is that essentially Denisovanes already developed the ability to live at high altitudes for some reason. And then when modern humans actually went outside of Africa and went into Asia, so the ancestors of Tibetans, they picked up this allele. And when they went to higher altitude, it was actually positively selected so that most individuals now carry this Denisova ancestry for this particular variant. So that would be a clear case of something that was advantageous. Another interesting study by colleagues from Leipzig has to do with the immune system. So there are these toll-like receptors that have to do with innate immunity. When you look at one particular cluster of 3 genes, that is in just one region, what you can actually see is that there are actually 2 different Neanderthal variants and 1 Denisova variant at exactly this particular region. And so when you look at the Neanderthal variant and you ask whether you actually see a gene expression difference compared to other variants, the African variants or others, you actually do see an expression difference. And this expression difference might actually translate in difference in how you can actually fend off certain viruses. So this would actually argue that when humans went outside of Africa, they also had to maybe adapt to different viruses and actually had the chance to pick up alleles that were advantageous in fending that off. In a more detailed study, this was also then tested by looking at eQTLs that you find when you stimulate certain cell types by infecting them with the flu or some viruses, and this again found specific regions in the genome that react to this and that are associated with it, and some of them actually come from Neanderthals. So there seems to be something that was actually advantageous by picking that up. So in summary on this selection story, it seems that there are essentially all 3 of these categories somehow filled. So there are some advantageous things that we picked up from Neanderthals, like altitude adaptation, and there seems to be also a role that they play in how our immune system works. There's certainly a lot of the variants that we carry right now seems to be neutral, so it might not really matter whether you're Neanderthal or not, for your phenotype or for selection. And there's also negative selection going on some regions, or was going on in the past, potentially because of some fertility issues, and also probably because some of the alleles were just not fitting the modern human background. So one thing that I actually want to bring at the very end is one particular variant that might be very interesting for this audience. So there was this one sequence variant that was found in Mexico that contributes to the higher incidence of diabetes in this population. And this particular variant in the gene SLC16A11 is actually from Neanderthals. So this is another case that's quite interesting. It's also interesting to see that this variant is actually relatively frequent in this population, but it's not so frequent that we can actually make an argument about whether it was positive in the past or not. And with this, I would like to sum up. So I talked about two different things here. So the first one is to study the phenotypes that we developed in the past in our own ancestors, and how we can actually prioritize which regions to study for that. And the second part was what Neanderthals, when they admixed with us, what kind of alleles they gave us, and what influence these alleles have for us right now. And I would like to end with acknowledgements. So the first part was driven mainly by Stefan Perenya, who is a PhD student in my group. And I had a lot of input from Micha Dannemann and Janet Kelso, Michael Lachmann, who was one of the persons that had the initial ideas and the initial design behind this test. And of course, lots of support from Svante Pebo, who leads this lab in Leipzig. And what I should also mention, without reading all the names, is that I had the pleasure to work with lots of different people on these analysis of different Neanderthal and Denisova genomes, and I'm highly indebted in their contribution to this analysis. And I thank you for listening. Thank you.